Hi everyone, this is going to be the second video in the sequence concerning biochemistry and molecular biology. In the previous video we looked at an overview of the idea of monomers and polymers in biology, and we looked very briefly at carbohydrates as well as other classes of biological polymers such as proteins and nucleic acids. In this lesson we're going to be focusing specifically on carbohydrates and looking at them in a lot more detail. So carbohydrates are incredibly important, not just in biology but also in the commercial context. They tend to be molecules which are used as a source of energy in organisms as well as structural materials in various parts of organisms such as membranes, cell walls, and as we saw in the last video, in the exoskeletons of many arthropods and elsewhere. Now, all carbohydrates contain the elements carbon, hydrogen and oxygen, and in carbohydrates the elements hydrogen and oxygen are present in a 2:1 ratio. So as we saw before, the general formula of a carbohydrate can be represented like this, where x and y are integers. So one example that we saw in the previous video was glucose, which hopefully by now you know the formula is, the molecular formula for glucose is C6H12O6, and so of course this conforms to that general formula that you're seeing here. But there's also disaccharides such as sucrose. Now sucrose is the typical granulated table sugar that you find in everyone's kitchens, and that has the formula C12H22O11, and this also conforms to this general formula for carbohydrates as well. So carbohydrates are typically classified as either sugars or polysaccharides, and sugars tend to be subcategorized as either being made up of individual sugar subunits, monosaccharides, or being disaccharides, that is, molecules composed of two monomer subunits, two sugar units. So carbohydrates can generally be classified like this, and then these can then be subcategorized as follows. So sugars can either be monosaccharides or disaccharides, and polysaccharides are typically classified according to their function. They can either be storage molecules or structural molecules. So monosaccharides are single sugar subunits that or units that can include things like glucose, fructose, and galactose. And so this is an example of one such molecule, glucose. We saw the molecular model for that, the ball and stick model, as well as the space-filling model in the last lesson. Disaccharides are double sugar units. These include things like sucrose, maltose, and lactose. Now, storage polysaccharides are categorized as storage based upon the fact that they're used by cells as long-term energy storage to store monosaccharide units in as small a space as possible. And so things like glycogen and starch are examples of storage carbohydrates. And so animal cells will use glycogen, which is a very dense store of glucose molecules, and the equivalent polysaccharide in plant cells is starch. Now, in terms of structural polysaccharides, a couple of the examples that we mentioned in the previous lesson are cellulose and chitin. These are both important structural polysaccharides, cellulose forming the fabric of plant cell walls, but also chitin is a component of the exoskeleton of many arthropods as well as the cell walls of fungi. So let's have a closer look at monosaccharides now. So these are the single sugar units that form the building blocks for larger carbohydrates. So in that respect, they're referred to as the monomers of the larger polysaccharide polymers. So there are many different monosaccharides. They vary according to the number of carbon atoms that they possess, and they tend to be referred to as such. So a couple of the examples that we saw in the previous lesson are hexose sugars, which contain 6 carbon atoms, so an example of this would be glucose. But you can also have pentose sugars such as ribose, which is found in nucleotides. Now, glucose exists as both a straight chain and a ring form, and the rings tend to form when glucose is found in aqueous solution. So here are the chain and the ring forms of glucose, and they both have the same molecular formulae, but two different structural formulae, and so these would be referred to as isomers of glucose. So taking a closer look at glucose then, so this straight chain representation of the glucose molecule shows how the carbon atoms are arranged and also how they're numbered. And so the first carbon by convention is the carbonyl carbon here. So for those of you who perhaps aren't studying advanced level chemistry, this functional group here, the carbon double bonded to an oxygen, this is referred to as a carbonyl group, and the carbonyl group is found in a number of other functional groups, including aldehydes, ketones, carboxylic acids, esters, and so on. Now, this end of the molecule, this carbon double bonded to this oxygen and this hydrogen, collectively these three atoms in this arrangement would be referred to as an aldehyde functional group. And so by convention, in the straight chain form of glucose, this carbon atom here is numbered carbon 1. And so glucose, in common with many other hexose sugars or 6-carbon sugars, has an aldehyde group as part of its linear structure. So this would be the aldehyde group here, and the carbon atom that forms part of this aldehyde group is always carbon 1. Now the carbonyl group itself, the carbon double bonded to the oxygen, has a number of important chemical properties, among which is its reducing properties. And so this is what makes all monosaccharides reducing sugars. So you may see them referred to as reducing sugars, and we'll look a little bit more closely at that a little bit later on. Now, the remainder of the molecule is a series of bonded carbon atoms with attached hydrogen atoms and hydroxyl groups. And so, again, the carbon atom of the carbonyl group is referred to as carbon 1, but sometimes it's referred to as the anomeric carbon atom. And so, again, for glucose, this is carbon 1. Now, in aqueous solution, glucose exists in a ring form that looks like this. So, glucose forms a 6-membered ring. Now, this is not a 6-carbon ring. The 6th carbon of glucose is this carbon here, but glucose forms a 6-membered ring in this manner. So, the hydroxyl group on carbon 5 bonds to the aldehyde on carbon 1 and forms this ether linkage that we see here. So the ring structure of glucose is usually represented, as we saw in the previous lesson, by a diagram referred to as the Howarth projection. And so this is given here. And typically these projections can also be better understood by building molecular models. Now, many of you will have done this in either your chemistry or biology classes. You may well have built molecular models of various molecules with these. Now, the advantage of building these molecular models or using software like you saw in the previous lesson that I was using to build 3-dimensional models of things like the glucose and glycine molecules, for example, is that in addition to looking at these things in a kind of skeletal or structural formula format on paper or a Howarth projection, you can see the actual way that the bonds arrange themselves in 3 dimensions relative to each other. And so the relative positions of the atoms in molecules can be demonstrated through building molecular models. So I mentioned isomers before. Now each hexose sugar, each 6-carbon sugar, can exist in 2 different forms, the alpha and the beta forms. And so again here we see alpha glucose and beta glucose. Now once again these are also isomers of glucose. Now an isomer is defined as, or rather isomers are defined as substances composed of molecules that have the same molecular formulae, in other words, the same ratio of atoms of each element present in each molecule, but a different spatial arrangement or different connectivity between those atoms in 3 dimensions. And so here we can see that alpha glucose and beta glucose differ from each other in the orientation of the carbon to hydrogen bond and the carbon to oxygen bond. In other words, the orientation of this hydroxyl group versus this hydrogen atom on carbon 1 of the 6-membered ring. So we see here in the Howarth projection of alpha glucose, we see that the orientation of those 2 bonds is opposite to the orientation of those same 2 bonds on the same carbon, carbon 1, in beta glucose. So we can use semi-skeletal, semi-structural formulae like these, and these can be represented in a number of different ways. And so the same 2 molecules can be represented like this, which is essentially a simplified view of the alpha and the beta glucose molecules. Now, those of you who perhaps aren't doing advanced level chemistry may be a little bit less familiar with these skeletal formulae, but a skeletal formula is essentially a kind of organic chemistry shorthand. It's a way of drawing organic molecules without having to draw every single carbon and every single carbon to hydrogen bond. And so, for example, everywhere you see a straight single line in one of these skeletal formulae, what you're looking at is a single covalent bond. If there is no atom at the intersection of these bonds, it's assumed that that atom is a carbon atom. And so we see here that there is no symbol for an atom here, but then it is assumed by convention of this notation that this is a carbon atom. And so here we can see 4 single bonds, which again makes sense if you're dealing with a carbon atom there. Here we can see that there are 2 single covalent bonds coming from whatever atom is here, but again we have not put a symbol for the atom because it's assumed that first of all that this is a carbon atom and that we have not drawn the additional 2 single bonds which we know must be there because those bonds are to hydrogen. And so the general convention is when you're drawing a skeletal formula, you don't bother to draw every single carbon to hydrogen atom and you don't bother to draw every single carbon to carbon bond. Instead, you simply draw single lines or double lines or triple lines to represent single carbon to carbon, double carbon to carbon, and triple carbon to carbon bonds respectively. And the carbons that you draw at these intersections between bonds, you don't bother drawing the symbol for carbon because it's understood that there's a carbon atom there. And similarly, if you see a single line here and a single line here, by convention, you know that this represents a carbon-to-carbon single bond, this also represents a carbon-to-carbon single bond, and therefore, if there are 4 single bonds around the carbon, the remaining 2 bonds which are not drawn are to hydrogens. So a disaccharide is a sugar molecule composed of 2 monosaccharide subunits covalently bonded together by a glycosidic linkage. So maltose, for example, which is also known as malt sugar, is formed from 2 glucose molecules joined by a glycosidic bond. Lactose, which is the sugar found in milk, is a disaccharide formed between the monosaccharides glucose and galactose, again linked by a glycosidic bond. And sucrose, which is common household sugar, is formed when you take the monosaccharides glucose and fructose and bond them together with a glycosidic bond. And so we form the disaccharides, maltose, lactose and sucrose, by taking one molecule of glucose and bonding it with another molecule of a different monosaccharide. And again, the bonds which are formed here are formed by means of a condensation reaction. So let's have a look at an example of one of these. So these are the Haworth projections of 2 alpha glucose molecules. Now, when we form a glycosidic bond between these two, we are essentially removing a water molecule, and so this is called a condensation reaction, and the bond that then results is referred to as a 1,4 alpha glycosidic bond. So, maltose is referred to as a disaccharide, and it's formed when 2 alpha glucose molecules are covalently bonded together by means of a condensation reaction, which produces a 1,4 glycosidic bond. Now, it's called a 1,4 glycosidic bond because it's from carbon 1 of one glucose molecule, and it's forming a bond via this oxygen atom to carbon 4 of the adjacent glucose molecule. So once again, for each one of these molecules, the anomeric carbon, the carbon 1 is here, and so this is carbon 2 here, carbon 3 is here, carbon 4 is here, carbon 5 is here, and this is carbon 6. So you need to be aware of how we can chemically test for the presence of reducing sugars. Now, all of the monosaccharides and many of the disaccharides are classified as reducing sugars, and we use a chemical test referred to as Benedicts Test to determine the reducing properties of the different sugars and their presence in a particular solution. So Benedict's solution is a turquoise blue coloured solution, and it contains copper ions and sodium hydroxide. Now, the copper ion exists as copper 2 in this reagent, so Cu2+. And so if a sugar is a reducing sugar, then the Cu2+ ions are reduced to Cu+. Now again, this goes back to the definition of reducing or reduction in chemistry. Reduction in chemistry can be defined essentially in 3 different ways. Oxidation and reduction have three different definitions in chemistry. So the first of these is probably the definition that you're most familiar with from GCSE chemistry, where reduction means the gain of electrons. So when a molecule is being reduced, it's gaining electrons from some other particle. Oxidation is the opposite. Oxidation means the loss of electrons. Now, there are two other definitions. The first, again, should be familiar to you. Oxidation can be defined as the gain of oxygen atoms by something. And so, for example, burning or combustion is an example of an oxidation reaction where a fuel is having oxygen atoms added chemically to it. And obviously the opposite is reduction. So reduction would mean the removal of oxygen atoms from something. But the third definition is perhaps the one that you're least familiar with at this stage, where reduction is the gain of hydrogen atoms by something, and oxidation is the loss of hydrogen atoms by something. Now, in this case, the copper 2 ion, Cu2+, is gaining an electron and is therefore being reduced to Cu+. So in the presence of alkaline sodium hydroxide, this then forms copper oxide. Now, the outcome of that is that the copper oxide is an insoluble substance, and so therefore precipitates out of solution, and it forms a brick red coloured precipitate. And so this is how we can test for the presence of reducing sugars. We start with a solution containing equal quantities of a food solution, which we're testing for the presence of reducing sugars, and Benedict's solution. Now, these two equal quantities of solution are mixed together and placed in a boiling tube, like is being shown here, and then this boiling tube is heated in a water bath, as shown here, for several minutes. Now, at the end of that, we see a change of colour through the green and then yellow to form eventually a brick red precipitate. And so this indicates the presence of a reducing sugar, such as, for example, glucose. Now, the brick red colour is the definitive result, which indicates the presence of a reducing sugar. So when Benedict's test is performed with disaccharides, such as maltose and sucrose, we get the following results. So, with sucrose, we get this result where the solution remains this transparent blue colour, but with maltose, we get the precipitate forming as a suspension in the solution, forming this colour here. And so from these results, we can see that sucrose is a non-reducing sugar, whereas maltose is a reducing sugar. So, in order to determine if the sucrose is present then in a sample or a solution, it's not enough to simply run Benedict's test on that solution. We then need to perform an additional test. So the sample or the solution under consideration is first boiled in hydrochloric acid, and it's the boiling in acid that breaks the glycosidic bonds. And so under these conditions, the glycosidic bond is hydrolyzed, and this procedure is called acid hydrolysis for that reason. The solution that you then form is then neutralized by adding drops of alkali while testing with pH paper or with a pH meter. And now that you've got a neutral solution, you can then perform Benedict's test on the resulting solution. Now, if you get a brick red precipitate forming, then sucrose was present in the original solution. Now, the reason for this is that the acid hydrolysis breaks the glycosidic bonds in the sucrose molecules, and this releases the free glucose and fructose monosaccharides into solution. Now, glucose and fructose are both monosaccharides and therefore both reducing sugars, and so it's for this reason that the subsequent test with Benedict's solution will yield that definitive result, that brick red precipitate. Now, if you get no precipitate formed, then sucrose was not present in the original sample, and so the need to neutralize the solution following the acid hydrolysis is due to the fact that the Benedict's test requires an alkaline medium, and this is the reason why sodium hydroxide is present along with those copper 2 ions in Benedict's solution. So we've talked a little bit about monosaccharides and disaccharides. Let's have a closer look at polysaccharides now. So as we said before in the previous lesson on monomers and polymers, polysaccharides are large polymers of the monosaccharides. Now, unlike the monosaccharides and disaccharides, in other words, unlike sugars, polysaccharides tend to be either insoluble or will form colloidal suspensions in water. Now, the principal storage polysaccharides are starch and glycogen. And starch is a polymer of alpha glucose and is in fact a mixture of two different polysaccharides, amylose and amylopectin. So as we said before, starch is composed of a mixture of these two different types of polysaccharides. So amylose forms these long unbranched chains of glucose units, whereas the amylopectin forms a highly branched polymer made up of glucose monomers. So amylose is formed by condensation reactions, which bonds the alpha glucose molecules together in a long chain with these glycosidic bonds as being shown here. So the amylose chain, once formed, will then coil into a helix, as shown here. So the amylopectin, on the other hand, will form these branches. Now the branches are formed by glycosidic bonds between carbon 1 on one chain of glucose, on one polymer chain, and carbon 6 of one of the monomers in an adjacent chain of glucose molecules. And so the branch points will occur roughly every, or rather approximately at every 12th glucose along the straight chain that you see here. So the branch points again formed at these positions. Position 1, carbon 1, the anomeric carbon on this monomer of this chain, and carbon 6 of this monomer on this chain here. And so for this reason, this glycosidic bond would be referred to as a 1,6 alpha glycosidic bond. And so amylopectin consists of a branched structure. It's highly branched, and this makes and this branched molecule wraps itself around the amylose to form the final starch molecule. Now, this large insoluble polymer is an ideal storage, or rather an ideal way of storing lots of these individual glucose monomers for the purposes of long-term energy storage. Again, glucose is the primary fuel, if you like, for metabolism, for respiration. But in order to store lots of individual glucose molecules in a smaller space as possible within a cell, it makes sense not only to polymerize them together like by glycosidic bonds, but also to do so in this branched format, because that way you have lots of individual glucose molecules at the ends of these branches, which can be cleaved off using enzymes that break the glycosidic bonds at the terminal ends of these branches. So the more of these ends that you have, the easier it becomes to release large numbers of glucose molecules all at once into solution for use in respiration or for transport to other parts of the cell or for other functions. So, another test for the presence of starch, or another food test that you need to be familiar with in this context of carbohydrates, is the test for starch. So the iodine test for starch is something that you may have come across in GCSE biology. So when iodine in potassium iodide solution, as you see here, is added to a food solution or a substance containing starch, the iodine molecules will pack inside the amylose helix to give the characteristic blue-black colour. And so when the iodine reacts with the starch in this piece of potato, we get this blue-black colour developing where the iodine reacts with the starch. So this electron micrograph, you can see here the scale bar at 50 micrometers here. This shows starch grains inside plant cells. Now, the glycogen molecules are the animal equivalent of starch. It's often referred to as animal starch. So glycogen has the same overall structure as amylopectin, but there's significantly more branching in glycogen than you find even in amylopectin. And so you get more of these 1,6 alpha glycosidic bonds which form the branch points. So glucose tends to be stored for long-term storage in animals, either in liver tissue or in skeletal muscle. And so another form, or rather another type of polysaccharide would be the structural polysaccharide. So again, sugars are classified as being monosaccharides and disaccharides according to their structure, whereas polysaccharides tend to be classified according to their function, whether they are storage polysaccharides, or structural polysaccharides. So cellulose is one of the most important structural polysaccharides, and it's the major component of plant cell walls. And so this is a diagram of a section of a cellulose molecule, and you can see here from the Howarth projections of the individual monomers here that we have 1,4 alpha glycosidic bonds. And so cellulose is a polymer, but this time of beta glucose molecules. And so each glucose molecule here is also inverted with respect to its neighbour, as you can see here from the Howarth projections. Now, the orientation of the beta glucose units places many of the hydroxyl groups on either side of the molecule, as you can see here. And so you can get many parallel chains of beta glucose subunits, and each chain can form hydrogen bonds between adjacent hydroxyl groups of adjacent chains. And so this is part of the reason that cellulose can give cell walls of plants their kind of rigid structure. So here we can see adjacent chains next to each other, and we can see here a micrograph of a cell wall where we can see the individual cellulose fibers laid across each other, parallel to each other, and also in layers like this. So the bundles of parallel chains forming hydrogen bonds with each other create a molecule that confers rigidity and strength on the structures of which they form a part. And so these lines here indicate not covalent bonds, but hydrogen bonds between parallel chains of beta glucose polymers. So once again, hydrogen bonds are not the same as covalent bonds. Covalent bonds are chemical bonds. They are bonds between atoms. Now, chemical bonds are orders of magnitude stronger than intermolecular or physical bonds. Now, hydrogen bonds are intermolecular bonds or physical bonds, but they are the strongest kind of intermolecular bond. And if you have very, very long polymers made up of beta glucose, then you can have many of these hydrogen bonds between adjacent polymer chains. And so even though the individual hydrogen bonds are not as strong as the covalent bonds that hold these polymer molecules together, the cumulative effect of having so many of these hydrogen bonds between the adjacent polymer chains gives these structures a lot of rigidity and strength. So again, let's have a look at some information that takes us a little bit beyond the most A-level specifications, just to give you that additional depth and breadth of understanding that you're going to need to get those higher marks. So we mentioned that sucrose is a non-reducing sugar. So let's have a look at why this is the case. So this is a diagram of the structure of maltose, and to compare, this here is a diagram of the structure of sucrose. Now, sugars will reduce Benedict's solution when the anomeric carbon atom is made available to reduce the copper ions in the solution. Now, the anomeric carbon atom is the carbon of the carbonyl group present in the straight chain form of the sugar, as we can see here. So once again, in the straight chain form, the anomeric carbon is the carbon atom found in this aldehyde group. In other words, the carbon which forms the carbonyl bond that you see here. Now, the anomeric carbon for fructose is actually carbon 2. So this is the straight chain form of fructose, and you can see here the anomeric carbon is this carbonyl atom here. And so fructose is a ketose sugar because this configuration of a carbonyl group in between two carbons within the main chain of a molecule, for example here, is not referred to as an aldehyde as you saw in the glucose molecule. This is called a ketone functional group. And so fructose being a ketose sugar, this will bond to glucose to form sucrose. And so this is the manner in which glucose and fructose bond with the glycosidic bond. And as you can see here, the anomeric carbon on glucose, or the carbon 1, is bonded to carbon 2 here. And so this is referred to as a ketone group. So when maltose is boiled with Benedict's reagent, the region of the ring containing the anomeric carbon, carbon 1, can open and expose the carbonyl group and this is capable of reducing Benedict's reagents. It's capable of transferring an electron to that copper 2 ion to form a copper 1 ion, and this is what leads ultimately to that change in colour and the formation of the precipitate, which indicates a reducing sugar. Now, only an anomeric carbon atom that is not involved in the formation of a glycosidic bond can be exposed in this way. Now, this potential anomeric carbon atom is available to reduce Benedict's reagent in the case of maltose. This potential anomeric carbon, on the other hand, is unavailable. So the one available anomeric carbon atom is sufficient for this molecule to reduce Benedict's solution, and thus maltose is therefore a reducing sugar. Now, if we contrast that with sucrose, sucrose is synthesized when glucose forms a glycosidic bond with fructose in this manner. And so this here would be the glycosidic bond that joins these two monosaccharide subunits together to form the disaccharide. And so the anomeric carbon for fructose is carbon 2. And so the anomeric carbon for glucose, again, to remind you, was carbon 1. And so, as both the anomeric carbon atoms are involved in forming the glycosidic bond when glucose and fructose join, there are no potentially free anomeric carbon atoms available to reduce Benedict's solution, and therefore sucrose is a non-reducing sugar for this reason. So, a little bit more information about structural polysaccharides. Now, again, we mentioned in the extension section of the previous lesson that chitin is a polysaccharide, and it forms the exoskeletons of insects, crustaceans, and also the fabric of fungal cell walls. Now, this is a linear polymer of a derivative of glucose called N-acetyl glucosamine. And so the N-acetyl is referring to this group of atoms here, linked to the second carbon in the six-membered ring here. So chitin will form the exoskeletons of both insects and crustaceans. So the interesting part of this is that crustaceans, insects, and also fungal cell walls have the same polymer, and largely for the same reasons, to confer rigidity and strength to their structures. Okay, everyone, that's going to do it for this lesson. I will see you in the next lesson, which will be all about lipids. See you then.